Hi there. In this video, I'd like to go through the Unit 3 notes, the first part of them that we're responsible for this week. So we're starting the skeletal system, and first thing up in the notes you can see in our book is just the basic functions. And so support, obviously of like soft tissue protection. I'm thinking things like the skull protecting the brain, the vertebral column protecting the heart and lungs, storage of minerals like calcium. Also, fat is stored in the medullary cavity. Movement, of course, as you know from lab, bones don't technically move on their own, but, and before you took this class, but what I mean there is like attached to the muscle. When muscle contracts, it creates directed movement because of the skeletal system. And then blood cell formation is another kind of funny one because they're referring to bone marrow, which is technically blood forming tissue, but it exists in the medullary cavity of bone, and because that blood forming tissue is housed by bone, they put it in as a function of bone. So bones are classified in two different ways. One way is based on their shape, and the other way is based on the texture. So I did make a video about these different types of classification systems, and it's very straightforward. And so I will show you that. I'll jump over to the course for a minute to show you the videos I'm recommending for this week. So functions of bone, like I said, pretty straightforward. And then, okay, so types of bone. Actually, I'm using somebody else's video that I found useful, and like I said, it's very straightforward because you either, and actually bone is characterized, just moving back to this, based on both at the same time. So it's kind of like, with epithelial tissue, how you can characterize it based on the shape, like squamous or cuboidal, and then you also classify it based on the number of layers, so it's simple squamous. So same thing here. Um, all of these bone shapes, long, short, flat, irregular, will all contain both types of bone. They all contain both compact and spongy bone. It's just a matter of where the compact is and where the spongy is. So this will make more sense as we go on in the notes today. So structure of a long bone, you go over in lab, and I posted a video on this, and so I'm not going to really belabor this, but the first thing that I go through is gross anatomy of bone. So the gross anatomy of a long bone would be the diaphysis, is that central, linear portion, and then the like bulging ends. Those are the epiphyses. The medullary cavity is the space in the middle of the diaphysis. So it's just a hollow cavity, and that's where the bone marrow is located. And we have two different types of bone marrow. We have red bone marrow and yellow bone marrow. Basically what that is it's active and inactive. So red bone marrow actively produces blood. That's primarily in the axial skeleton, whereas yellow bone marrow, it's inactive, and it's overrun with adipose tissue. And so red bone marrow, what I mean by axial skeleton, which you probably have heard this in lab, is like the central core of the body. So the sternum, um, the hip bones, the ribs, the proximal epiphyses of like the humerus and femur, all like very active in producing blood. But once you start going down the limbs, you see more yellow bone marrow. Now, once bone marrow converts to inactive yellow bone marrow, it can convert back. So if we had a need to produce more blood, then this yellow bone marrow could, revert back to red bone marrow and give rise to more blood. As it is in normal physiology, we don't have the need for all of that active bone marrow. So those are the basic parts of a bone. And then I tried to organize this for you. So there's cartilage in two locations. There's cartilage between the epiphysis and the diaphysis, and you're going to call that a different name based on the age of the bone, basically. So the epiphyseal plate, also known as the growth plate, is a thicker piece of cartilage in between the epiphysis and the diaphysis. Compared to once linear growth of the skeleton has ended, what you see is a remnant of cartilage there, and that's called the epiphyseal line. Okay, so that's in between the epiphysis and the diaphysis, and then capping the ends of long bone is the articular cartilage that's found making up a joint. So that's the two places that we see cartilage. And then we also see membrane in two places. And I put a little picture of that in our notes, and this is also in your book. But the periosteum, that's the membrane on the outer surface of bone, which we'll talk about again next week. So taking a look at this picture, what they're trying to show you here is they are, peeling back the periosteum with like forceps, which is unrealistic. It's really kind of like adhered to the surface of the bone. But what they're trying to show you is there's two portions of it. There's this outer layer. It's called the fibrous periosteum, which is really dense collagen, like connective tissue, very strong. But then inside of that, you might be able to tell they're drawing it more cellular, so you can see the cells in there. And this is known as the cellular layer of the periosteum. Okay, so the periosteum has these two layers, kind of a fibrous connective tissue coat, and then the inner cellular layer, which that intercellular layer has a lot to do with like growing the bone. So that's what we see on the outside, this double layered periosteum. And so an adjective that's used a lot is periosteal. So the periosteal surface is the outside of the bone compared to the endosteal surface. The endosteal surface is the inside of the bone. It's the lining of the medullary cavity. So this would be the open space here where the bone marrow is located. And here you can see the endosteum is made up of, various different cell types. But it's just cellular. It's not connective tissue. So there are some cells. This is a great big cell called an osteoclast. These are the osteoblasts. So this is a stem cell, osteoprogenitor cell. And then the osteocytes are the ones that are trapped within the bone. So I did make a video that goes over those different cell types in detail. So I'll just leave it at that overview. But basically, what we have here with the bone is the membrane at these two locations. Okay, so outside of the bone and lining the medullary cavity. Okay, all bones aren't long bones, so that's long bone anatomy. That's very important for us to know. But the other shapes of the bones, like short, flat, irregular bones, those are basically, I would call it like a spongy bone sandwich, where you're going to have compact bone on the outside and then it's filled with spongy bone, kind of like sponge candy, like the chocolate on the outside, that would be the compact bone, and then the sponge candy on the inside. That's what the short, flat, and irregular bones look like. And this video classification of bones, let's see, just to show you. Okay, so the long bone anatomy, I'm relying on somebody else's video. I know in our playlist this looks like it's not working. I clicked on it, it is working, so I'm not sure why the title sheet looks like that. But classification of bones, this is the video that I made for you that talks about the bone shapes and the spongy bone, and I show you, how that sponge candy type of arrangement. And so then what I do in the notes after, basically what I'm doing is I'm telling you how to classify bone, the shape and the texture, showing you the long bone anatomy, which this is very important. You're going to get this in lab and in lecture. It's very important also. And then the short, flat, the other shapes, that's pretty straightforward. So the I guess angle that I take here to look at bone in more detail, because we just finished connective tissue, is to look at bone as a connective tissue, because it is a connective tissue. And therefore it has cells spread out here and there, and then it has the non-living matrix in between those cells. And the matrix, of course, is the fiber and the ground substance. So because bone is a connective tissue, it contains all of these cell types, the stem cells, the osteoblasts that produce bone. They can produce the matrix of bone. That's known as osteoid. The osteocytes are the more mature cells. Those are the ones that trap themselves in a lacuna. The osteoclasts are completely different. Check this out. Derived from white blood cells. Completely different. If you know that white blood cells can phagocytize, can engulf foreign things, that's basically these osteoclasts look, they look very different and they act very different. They act as phagocytic cells and they actually break down bone. So they do the opposite of the osteoblast, one letter difference. So the osteoblast makes bone, and the osteoclast breaks it down. I gave a little picture here, but like I said, I made a video that goes through this in a lot of detail, so I think that you'll be happy with that. That's this video, bone as a connective tissue, cell types. So the fiber type that you find in bone is collagen, which weaves through the ground substance, which is just typical, believe it or not, proteoglycans and glycoproteins. So in other words, the ground substance is just protein and sugar, just like we learned in Unit 2. It has a thick, syrupy consistency, which is very different from, what we're thinking when we think of the matrix of bone. I'm thinking of something, that's solid, right? So what happens is there's no cell like the osteoblast that can make solid matrix. And so it makes the matrix as the syrupy, it's just like sugar and protein, deposits it around. And then what will happen is calcium and other minerals from our blood will actually precipitate out of solution and deposit in the ground substance. And that is what solidifies it. And so I talk about that a little bit in the notes, how calcium can exist in two states. It can be a liquid, right? And it's soluble in the blood. But then what can happen is it can crystallize, precipitate out of solution. That's what is known as a hydroxyapatite. Those are the calcium crystals that are found in the matrix of bone. And so the matrix of bone becomes solidified following the production of bone. So like I said, this video, bone as a connective tissue, I'll show you this one, is where I explain that in, more detail. So going on to the next topic and just kind of keeping track here, can you see how what we're doing is we're looking at the gross anatomy? What can I see with my bare eyes, right? The shape, the texture, the gross anatomy of a long bone, gross anatomy of the other shapes, what cells are there, what fibers are there, what is this tissue? And then I'm looking at the microscopic anatomy of bone. And they cover this in lab. And I made a video that's detailed about the microscopic structure of bone, this one. So I'm going to let you watch that, and also you'll get this in lab, and reach out to me if you have any questions on this microscopic appearance of bone. So compact bone has a lot of detail. Spongy bone is just basically these little spicules of matrix. They're not like true osteons. They don't have the central canal. So see what you think there. The next large topic, so actually after all of this is done, see if this makes sense. So we know how to classify bone, we know the gross anatomy, we know the microscopic anatomy, and so then for, like, I'll say the whole rest of the notes, what I do, in this chapter is take the approach of looking at like the lifespan of a bone. In other words, how is the bone formed? And then how does it grow? And then finally, how does it change throughout our life? That's the next three big topics in the notes, starting with, how bone develops. So bone develops in two different processes. And bone development is known as osteogenesis. It occurs during late embryonic and early fetal development. That's when we start producing bone. Okay, so like I said, 3 stages of the bone's life, and this is how my notes read out. First the bone has to form, then it has to grow, then it has to maintain itself. So I go through all of this week. So as far as bone development, there's 2 mechanisms by which bone develops, either intramembranous ossification or endochondral ossification. So I do have videos on those, but the video on intramembranous ossification is one that, well, actually both intramembranous and endochondral ossification, both of those are videos that I'm just linking to somebody else explaining it. So I'm going to take a few minutes now to explain those 2 processes so you can hear that in my words, and hopefully that helps you out. So basically, these 2 different processes form different bones, and it's the same from person to person. For instance, our skull, all of our skulls formed by intramembranous ossification compared to all of our long bones formed by endochondral ossification. So it is the same from person to person, but the big difference is that there are two precursor materials. So bone can't form out of the blue. It's not going to, our skeleton doesn't form out of nowhere. There's going to be a skeleton looking structure made out of fibrous membrane and hyaline cartilage early on in development as an embryo. And then what happens is bone formation occurs within the fibrous membrane and occurs within the hyaline cartilage. And those are the two processes by which bone forms. So intramembranous ossification, intramembranous, that's when bone forms from inside of a fibrous membrane. And a great example of that is the skull. And so you can see I try to go through this step by step, but I'm going to jump over to some pictures. So this picture I pulled up to share, I didn't want to forget this one, is a really interesting photo from a different book. It shows the fibula, and it just shows without collagen in the matrix, it wouldn't have any flexibility. It would just be all those precipitated crystals, so it's very hard. So you can see when it breaks, it shatters. We know that's not the case. Actually, the collagen fibers weave through and it creates like an infrastructure, almost like, like the fibers in like a windshield, so that it doesn't completely just, fall apart into shards. But if the bone doesn't have mineral, so if we don't have calcium deposit in the bone, but you do have collagen, you can see how soft the bone is. You can actually bend it. And so we need the calcium depositing. So back to our topic of intramembranous ossification. So in this picture, we're looking at fetal development, and you can see around the skull area, that white shadowy structure is a fibrous membrane, and I can see in orange the hyaline cartilage in the limbs. And so intramembranous ossification is the process that's going to replace this fibrous membrane by bone. So let's look at that first. So I'm using pictures from the book. I hope this makes sense. So at first, that fibrous membrane, what it is mesenchyme. Okay, so it's the embryonic connective tissue that we learned about in part 2 or unit 2. So the mesenchymal cells are these purple cells, and that tough fibrous membrane surrounding the developing brain is just this generic, connective tissue. But what happens is some of these mesenchymal cells, because remember, these are the ultimate stem cell, they can differentiate into any connective tissue cell type, and that's exactly what they do. Some of these cells will differentiate into the bone-forming cells called osteoblasts. And when the osteoblasts form, what they do is they produce the matrix of bone. They produce the collagen fibers, and they produce the sugar protein mix, the ground substance. And that's what they're depicting with these little yellow dots. And so what they're trying to show us is the osteoblasts, once they form, they do what osteoblasts do. And that is they produce the matrix of bone, they produce the osteoid, and they're secreting that osteoid all around the cells. Okay, and so that is a center of ossification within that fibrous membrane. It's turning into bone. So let's fast forward. So now in this picture, what they're doing is they're showing us how two of those osteoblasts, here are the two osteoblasts, and you can still see the mesenchymal membrane out here. Two of those osteoblasts secreted so much matrix. Do you see how they got trapped in the matrix? Now, because this osteoblast is trapped in the matrix, it's going to stop producing osteoid, even though that's the job of the osteoblast. So you can see the ones that aren't trapped, those are still osteoblasts. They're still going to produce matrix. But the one cell that is trapped, do you see how they're calling that an osteocyte? An osteocyte is an osteoblast that stopped producing osteoid, and it stopped producing osteoid because it trapped itself into that little area where it's going to live that we call a lacuna. So here is another osteocyte in a lacuna. Here's the matrix that's becoming solidified. I'll talk about that in a minute. And here are the osteoblasts, and they would keep laying down more osteoid in this area. And you can see bone growth is starting from this area, and it's radiating out into the mesenchyme. So it's kind of like the ripple in a pond. once bone starts forming in one area of the mesenchyme, it radiates out from that point. Now, as these cells produce osteoid, they also produce a waste product called alkaline phosphatase that gets carried away by the blood. And that waste product, the alkaline phosphatase, actually stimulates, it attracts calcium into the area. And so that's going to cause calcium to come into this area and deposit in this newly formed osteoid, and that's going to solidify it. And that's basically how intramembranous ossification works. Just moving forward in our pictures, blood vessels will infiltrate the areas of bone growth, and the bone can't grow over top of a blood vessel, it grows around the blood vessel. But as bone production occurs, what's going to happen is it keeps clamping down so that blood vessel takes up a smaller and smaller area. But basically, this is the formation of spongy bone. As you can see, the trabeculae and the blood vessels weaving through that area. Out here, this purple area, that is still mesenchyme. But instead of calling it mesenchyme, this is going to form the periosteum. And I'm going to fast forward this picture one more time. So the periosteum forms by this layer of osteoblasts that's still on the outer periphery. And then what was originally the mesenchyme, we would call this the fibrous periosteum. And this layer of osteoblasts, that's the fibrous periosteum. We have formation of spongy bone. The cells that are trapped are osteocytes. Taking a look at a slide, we don't look at this in lab, but this white and with the purple dots, that's mesenchyme. This pink area, that's bone formation. So you can see how bone formation starts and it radiates out from that point, and how blood vessels get trapped in that. And the cells, so here are the cells trapped in the lacunae, those are smaller. And then moving forward, you can see how those spicules turn into a solid area, can't make bone over blood vessel. Blood vessels are still weaving through there, and the osteocytes that got trapped in the lacunae. The forming periosteum, you can see the cellular layer, fibrous layer. And so just taking a look at the skull, this is the fetal skull. You'll talk about that in lab, but just taking a quick look at this, do you see how, like, when the frontal bone forms, it actually forms in this, it starts to form in the same areas, and then that bone growth radiates out from that point. So by the time the person is born, bone growth has not filled in that entire mesenchyme membrane. There's a big gap there that's called a fontanelle. So a fontanelle is just the original fibrous membrane, the mesenchyme that's encasing the brain, that we haven't fully formed the skull yet. And we're born with 6 fontanelles. 2 are on midline on the top, and 2 are on the lateral surfaces, so 2 on each side, making 6 total. And I believe you'll look at those in lab. But I hope that this intramembranous ossification description makes sense, and that you can see how fontanelles would, naturally be there. That's just the part that didn't get filled in with bone yet. So that is intramembranous ossification. So going back to the notes, what I did is and I'll let you read through this yourself, I wrote out these steps step by step. Okay, so hopefully, this makes sense, and if not, I'm here for you to answer any questions that you might have. Now, endochondral ossification is very, very similar. It's just that you're using a different precursor material, hyaline cartilage, and long bones form by a hyaline cartilage model, endo in chondral, chondrocyte cartilage. So inside a cartilage, endochondral ossification is when the bone replaces hyaline cartilage. And so I'm going to go back to pictures, and I'm just going to run through this mechanism for us. So taking a look at a picture from a different book. Let's start here. So with this picture, the blue bone shape is not actually bone. It's a hyaline cartilage model of what the bone will be. So this is how a long bone would form, that you'd have a hyaline cartilage, which was produced by mesenchyme. Okay, so the hyaline cartilage also came from mesenchyme. Mesenchyme gives rise to all connective tissues. So hyaline cartilage model was produced by mesenchyme. You can see blood vessel infiltration to the cartilage, which is very unusual because cartilage is avascular, just like epithelial tissue. No blood ever infiltrates the cartilage. So you know when blood vessels start to infiltrate this area, something big is going on. And what big is going on is that some of the cells within the perichondrium, what you call the fibrous coating on the cartilage bone, some of these cells are going to turn into osteoblasts. So some of these cells are going to differentiate into osteoblasts. And you know what osteoblasts do. This is the same in either process. They produce osteoid. Osteoblasts produce osteoid, and that's what this thin layer of yellow or brown is that they're laying down. It's not going to be able to go, penetrate into the cartilage. It's going to get laid right down on top of the cartilage, kind of like frosting on the cake. You know what I mean? It's just going to get a layer right on top. So once the osteoblasts form, start laying down osteoid, what happens is because the cartilage is avascular, you can see how these cartilage cells are starting to struggle. This is called hypertrophy. These cells start to enlarge because we're cutting off the diffusion that was occurring with the interstitial fluid by laying down a layer of tissue that's becoming ossified. So when you lay down that tissue, that's going to kill the cartilage. And if we jump to the next picture, you can see what will happen is, this area starts to die off as the cartilage starts to get built up on the outer periphery. And as it dies off, at the same time, blood vessels, of course, we know are infiltrating this area. Eventually, they make it into this area that's dying off, which is going to ultimately become the medullary cavity, and it brings in the bone marrow and the osteoclasts, right, that are derived from the white blood cell that will make that cavity bigger. So jumping ahead, here you can see the initial formation of the medullary cavity. So the cartilage model is dying out. Osteoid continues to be deposited on the diaphysis. And what will happen is calcium is deposited in the middle of the diaphysis, and that calcium deposit moves towards the epiphyses. This is what's known as primary ossification just because it occurs first. So this diaphysis formation is where you see the primary ossification center, which is just where it becomes calcified first, right in the middle of the diaphysis. The epiphyses start to become ossified later, prior to birth, but just later in time. So just going through the pictures, here's the next picture where it shows the blood vessel infiltration, and it's infiltrating all of this area too. It's not just a single vessel. I know that's a little deceiving, but that brings in the osteoclasts and the bone marrow. And jumping ahead to the next picture, here we can see that some of the mesenchymal cells actually start to differentiate within the ends of the epiphyses into osteoblasts. So it's the same story. The osteoblast secretes osteoid. As it secretes osteoid, it's so metabolically active, it's releasing this waste product called alkaline phosphatase that's going to encourage the deposit of calcium crystals, solidification of that matrix in the epiphyses. And this picture, would be what you'd see like late in development. But believe it or not, it's a variation of this when a person is born. And so taking a look at another picture that goes through the whole process. In this picture, they're showing us that around the time of birth, we might see secondary ossification in one or maybe both of the epiphyses. But you see how there's so much cartilage that's left? So this cartilage in between the diaphysis and this solid epiphysis, this is what we call the epiphyseal plate. That's the growth plate. Okay, so that's the epiphyseal plate. And then this cartilage that's on the outer periphery is the articular cartilage. And here they're fast forwarding to adolescence, and you can still see presence of that thick cartilage pad. That's the epiphyseal plate, and then the articular cartilage. So when you understand endochondral ossification, it makes sense, where that cartilage is coming from. So in the notes, same thing. I wrote out all the steps, so hopefully that makes sense. Now, as far as bone growth, I did make my own videos on this, and so I'm going to rely on those to keep this video shorter for you. So bone grows in two ways, basically. It can get longer, as long as there's an epiphyseal plate. And I talk about that in the video on longitudinal bone growth. So that's this video, and I mark up the picture that I put in your notes, and so you might like to have some colored pencils or something, and I go through that diagram and talk about longitudinal bone growth, and like I said, mark that up in your notes with me. And the other way that the bone, so here's the picture that I mark up in that video, the other way that the bone grows is it can grow wider, and that's appositional bone growth. And I did make a video about that, and you can see I use that diagram that's in the notes, and you can mark up that diagram with me. Which brings me to the last topic, which is maintenance over time. So this is a nice, straightforward video that goes through that. But because this is, I think, possibly one of the more difficult topics, I'm going to go through that now also. So we formed the bone, right, by two processes. Then we grew the bone longer, wider. And now the maintenance of the bone just has to do with, believe it or not, calcium levels. So the question of where you're going to build up bone is a question that I'll talk about next week. I'll start with that next week. But why? Why would you build up bone, or why would you break down bone? Believe it or not, it's hormonally regulated, and it has to do with just regulating our blood calcium level. And so it's entirely dependent on blood calcium level. In other words, I'm not building up bone unless I have the calcium to do it. Or if I'm running low on calcium, if I have low dietary calcium, I'm going to take that calcium from my bones and put it in the blood, and to maintain my blood calcium levels. And that is because calcium is extremely important. It's needed for conduction of nerve impulses, muscle contraction, coagulation. So you can see these are big processes. Mitosis requires calcium. And so we monitor our blood calcium levels. And if the blood calcium level goes too low or too high, we correct it. So the way that I like to teach calcium homeostasis or maintenance of the bones in my face-to-face class is using the picture that I put in your notes on page 7. So let's look at this picture together, and I'm going to mark it up. So what they're doing in this picture is they're using a teeter-totter analogy. I'm going to look at the first half, the top half first. And so what they're doing is they're saying, okay, when we're in perfect balance, that our blood calcium levels are between 9 and 11 milligrams per 100 mils of blood. Okay, so they're just giving us the normal range. We don't have to memorize the normal range. The point here is if we go above that, so if more calcium is in our blood or calcium comes out of our blood, we're going to use hormones to fix that. That's how important it is to us. So again, calcium deposit in the bone, think of the bone as like the bank of calcium. If you need calcium in your blood, you're going to withdraw it from the bone. If you want to, if you have excess calcium, we can deposit it in the bone and build up bone. So that's the basic way this works. So in this scenario, here they have a rising level of blood calcium. So blood calcium level is going up. That would be what I call the stimulus. Okay, so that's the stimulus that's going to trigger something to happen, right? And so if we had a high blood calcium level, why would that be? The most likely reason is because you just drank milk or took a calcium supplement or something like that. I bet this is, dietary. That's probably typically what happens, that when we ingest something with calcium, that calcium enters into our blood, so our blood calcium levels go up. And it's the thyroid gland that monitors our blood calcium levels. So the thyroid gland, we'll talk about it again in AP2, butterfly-shaped gland right in the voice box area. that contains mainly follicles. And believe it or not, you've already looked at follicles in lab when you looked at simple cuboidal epithelial tissue. So remember how you had those spherical, or actually, on the slide, it's just round structures, and those round structures had a single layer of cube-shaped cells. So in the thyroid, this is what it looks like under the microscope, regardless of where you look. It's very uniform. And those parts of the thyroid have to do with the hormones that regulate metabolism. So I'm sure you've heard how the thyroid gland regulates metabolism. So in other words, they have nothing to do with calcium, but there are cells in between the follicles, and those cells are called parafollicular cells or C cells. And those are the cells that are going to respond to that high blood calcium. So if we have an increased level of calcium, it's the thyroid gland that recognizes that. The C cells release a hormone called calcitonin. Calcitonin is a hormone, it's released into the blood, and it targets the bone. Because remember, if we have excess calcium, I'm going to deposit it in the bone. And so it targets the bone, and what it does is it stimulates calcium deposit in the bone by stimulating the cells that make bone, which I hope you remember are the osteoblasts, right? So calcitonin is going to stimulate the osteoblasts to start making new osteoid. New osteoid is deposited on the surface of the bone, and then calcium will deposit in that newly formed osteoid. And when the calcium deposits there, that's going to bring our blood calcium level back down. So that's the effect of the hormone. So this scenario with calcitonin starts with C, I would think of it as being, more important in childhood, because in childhood, that's when the skeleton has to form, and we encourage kids to have high calcium foods because that high calcium then is able to deposit in the skeleton and form the skeleton. So I hope that half of it makes sense. That's if we have excess calcium. So now to the other side of this story. I'm going to erase these notes so I can move my picture. Okay, so here's the other part of the scenario. So another possibility is our blood calcium level is not within the normal range, that it falls below the normal range. Because you haven't had anything in calcium, or you have a low calcium diet, or we didn't take the supplement. And as an adult, this is definitely going to be the primary scenario that, not like chronically necessarily, but here and there, our blood calcium levels will fall below where they should be. So again, what's monitoring the blood calcium level is a gland that's associated with the thyroid gland. Now here we're looking at the posterior surface of the thyroid gland, the back of it, and they're showing us these little nodules embedded in the back of the thyroid, which is a completely different gland called the parathyroid glands. So the parathyroid glands are the ones that are sensitive to this stimulus of a low blood calcium level. And when the parathyroid glands sense that there's a low blood calcium level, they release the hormone parathyroid hormone. That's the only hormone that's released by the parathyroid glands. Parathyroid hormone is also going to target the bone, but this time this hormone targets the osteoclast. And what the osteoclast will do is degrade the bone, break down the bone, right? And by breaking down the bone, it's going to release calcium into the blood. And so that's where we're getting that calcium. If we have low calcium in the blood, we're going to withdraw it from the bone. The osteoclasts work on the endosteal surface. So when we break down bone, we make the medullary cavity bigger. We don't change the, outside shape of the bone. And so the effect of parathyroid hormone is to increase our blood calcium level. Back into the normal range. So I hope that makes sense. That's one of the toughest things in this whole unit, I would say. And so what I did in the notes is I wrote out the scenario too. So if blood calcium levels are too high, this is what happens. So that's what we went through on the picture, the top of the teeter-totter. If they're too low, here's the mechanism that occurs, and then this chart I left for you to practice. So see if you can fill out this chart all, on your own without looking at notes or anything. So if you can fill out this chart, I think that, you know, that you're doing great, that you basically got it. If you have questions, please reach out and we'll pick up from here next week. Thank you.